Hello and welcome, Biology 202 students. Welcome to your online lecture. My name is Chris Groberg, and I'm excited to be your online lecturer this semester. Today, we'll dive into the syllabus, discuss the course structure, explain how you'll be evaluated, and outline what to expect for the first week of lab. Let's begin this journey into the fascinating human anatomy and physiology world. For today's agenda, we're going to discuss the structures of a hybrid class, also how to succeed in this class. We will do an anatomical positions and planes review, then we will go over the tissues that you need to know. We will review how to use a microscope, and we will summarize appropriate dissection etiquette. This hybrid format will allow you to experience a blend of online lectures and hands-on lab sessions. This approach will enable you to learn independently, review complex topics as needed while gaining practical experience crucial to mastering anatomy and physiology. You'll watch these lectures each week before attending your lab sessions. This format ensures that you come to lab prepared, allowing us to maximize our time with hands-on activities, discussions, and reviews. In human anatomy, our main goal is to understand how the body systems work together. But first, we got to break down the body systems and learn about them individually. So, the first semester of anatomy, we talk about the integumentary system. That's the hair, the skin, and the nails. Then we go into the skeletal system. This is all of the bones and the structures within each bone. Then we hit the muscular system, which talks about how flexing muscles moves different parts of the body and how muscles work together and how they work against each other. Then we get into the nervous system, where we talk about the nerves in the body, how they function, and the brain. In the second semester, we talk about a few more body systems, but not to the intense degree as the ones in the first semester. So we talk about the endocrine system, the blood, the heart, the arteries, the veins, the lymphatic system, the respiratory system, the urinary system, digestive system, and the male and female reproductive systems. To succeed in this course, you will need to prepare for each class, participate in class activities, and have consistent study habits. Here are some key strategies. Watch the lectures. Watch all the online lectures before coming to lab. Take detailed notes to help reinforce the material. An example of this would be to watch the online lecture for Lab 1 before coming to class. This way you'll be ready for the activities, you will have good questions to ask, and the class should go more smoothly. Engage in lab activities. Actively participate in all lab activities. These hands-on experiences are vital for understanding the material. Review regularly. Review your notes and the lab manual regularly. You can create flashcards, participate in study groups, and use hands-on aids like the models in class. Within each module, we will have resources for you to look at. Complete the assignments on time. Ensure all assignments are completed on time. Use the syllabus to keep track of due dates. Communicate. Contact your TA if you have any questions or need clarifications. We're here to help. Lastly, read the syllabus. All the due dates are in the syllabus. All expectations are in the syllabus. If you read this and then add the due dates to your calendar or planner, you will do well. Okay, now let's review anatomical position. Understanding the anatomical position is fundamental to studying human anatomy. The anatomical position is the body being upright, facing forward, with arms at the side, palms facing forward, and feet flat on the ground. This standardized position is essential because it provides a consistent frame of reference when discussing different parts of the body. Why is this important? Well, it ensures that descriptions of the body are unambiguous, which is crucial when communicating with other healthcare professionals. Practice. Stand up and position yourself as described. Visualize this position when studying anatomical structures to understand their location and relationships better. Now let's talk about anatomical planes. So these are imaginary lines used to divide the body into sections, which helps us describe locations and movements. First, we have the coronal plane. The coronal plane, also known as the frontal plane, divides the body into anterior, meaning front, and posterior, meaning back. Then we have the sagittal plane. The sagittal plane splits the body left and right. The mid-sagittal plane divides the body into equally left and right halves. So you can have the mid-sagittal plane showing equal halves, or just a sagittal plane that's not exactly split 50-50. We also have a transverse plane. The transverse plane divides the body into superior, meaning upper, and inferior, meaning lower parts. Understanding these planes is crucial for describing movements and locations of different parts of the body. There's one other plane, and this is one that doesn't fit into coronal, sagittal, or transverse, and this is the oblique plane. The oblique plane is something that doesn't align with any of these. Now let's cover some basic anatomical terminology. These terms help us describe the position and relationships of different parts of the body. We have superior, meaning above or towards the head. Inferior, meaning below or towards the feet. Then we have anterior, meaning the front, and posterior, meaning the back. There's also dorsal, meaning posterior, and caudal, meaning anterior. So there's a few words to describe each of these. So we can draw a midline. This would be along the mid-sagittal plane, and this will split the body perfectly in half, 50-50, left and right. You can see here a green lateral arrow and a red medial arrow. Medial means towards the midline of the body. Lateral means away from the midline of the body. Then we have proximal and distal. Proximal means closer to the point of attachment or to the trunk of the body. Distal means further away from the point of attachment or the trunk of the body. Now this can be a relative term too, because you can say proximal and distal by themselves and it means that, or you could say proximal to something. So the thumb is proximal to the hand while the elbow is distal to the hand. Now it's time for a little tissue review. Tissues are groups of cells that perform specific functions. There are 4 basic types of tissue in the body. There's epithelial tissue. This covers the body's surfaces and lines cavities. The functions include protection, absorption, and secretion. Then there's connective tissues. These support and bind other tissues. This includes bones, blood, and fat tissues. We also have muscle tissue, responsible for movement. Types include skeletal muscle, cardiac, and smooth muscle. Lastly, we have nervous tissue. This transmits electrical impulses. It is made up of neurons and supporting cells. Okay, let's begin. So first I'm going to tell you a little bit about this tissue, and let's see if you can guess what kind of tissue it is. So let's start off with the appearance. So it's composed of neurons with large prominent nuclei and glial cells, which are smaller and less distinct. Some histological features. Look for neurons with dendrites and axons, and supportive glial cells. Neurons may be stained to show long processes, while synaptic connections are sometimes visible with special stains. Do you know what it is yet? Let's take a look at the locations. We find this in the brain, the spinal cord, and the peripheral nerves. That's right, this is nervous tissue. Alright, now let's do the same thing with these tissues. We'll start with some histological features on the left. There are striations that are parallel lines with fibers. You see nuclei that are flat and pushed to the edges. You see long cylindrical fibers with obvious striations and multiple peripherally located nuclei. we find these attached to bones. What do you think it is? That's right, skeletal muscle. Let's go to the next one in the middle. So this will be a little more obvious once I start showing you the histological features. We have intercalated discs. These are distinctive for their perpendicular lines across the fibers, facilitating synchronization of heart contractions. Can you guess? Yep, it's cardiac muscle. And then the last one on the right. Let's start off with the locations. So on the right, this is found on the walls of hollow organs, such as the intestines and blood vessels. The appearance is spindle-shaped cells with central nucleus, no striations, and histological features are, well, they're cells that are organized in sheets. Nuclei appear elongated and central, and it is smooth muscle. That's right. All right, we have 3 new tissues on the screen, and let's start off with the location. So on the far left, we'll start with that one. We find this in tendons and ligaments. You see tightly packed parallel collagen fibers with few cells, and the cells that you do see are primarily fibroblasts. For histological features, you see fibers that appear wavy due to tension. Nuclei of fibroblasts are squeezed between collagen bundles. So what do you think this is? That's right, dense, regular connective tissue. Let's move to the next one. So we find this in the dermis of the skin and organ capsules. It appears thick and has irregularly arranged collagen fibers with interspersed fibroblasts. Histological features, we lack the organized pattern, so it's pretty irregular. And it provides strength in multiple directions. So, what would you think this is? Dense, irregular tissue, that's right. And last but not least, we have our tissue on the right. We find this on the walls of large arteries and certain ligaments. It appears predominantly with dark staining elastic fibers that are wavy, and they stretch, which allow recoiling. So, what would you say this is? Yep, elastic connective tissue. I'm just guessing you're getting these right. But if you're not, that's fine. You can pause the video, go back, and practice these. Okay, so now let's move on. We have 3 images here. I'll give you a hint. They're all cartilage. This is, we're going to start on the left, and this is found in the trachea, larynx, and along the end of long bones, and also between the ribs and sternum. You can see some chondrocytes there. It looks a little more transparent, and that's because we can't really see the type 2 collagen fibers there. So, this is hyaline cartilage. Okay, and then let's move to this middle image here. This is another type of cartilage. There's a label here that gives it away. So this is fibrocartilage, and it's found mainly between these bones. These are vertebral bones. So in the intervertebral discs, you'll see fibrocartilage, and it's really good at resisting compression. And on the right here, you see very dark staining fibers. You still see chondrocytes. And what you really need to know about this is it's found in the ear, the pinna of the ear. You also see it in the epiglottis and the larynx. It's very similar to hyaline cartilage, but it has a lot of elastic fibers in it. So we call this elastic cartilage. Okay, we've covered a few things now. You can pause the video and stretch your legs, do a few jumping jacks, and then you can press play, and I will be here waiting. All right, go ahead and do that. Welcome back. I hope you did it, but also if you didn't, that's also fine. So let's start again on the far left with this tissue. This tissue serves as the body's major energy reserve, and it provides insulation and cushioning to the organs and tissues. It also plays a crucial role in metabolism regulation. It's found beneath the skin, so the subcutaneous area, and it's around internal organs, so visceral area, and in bone marrow. It appears as composed of tightly packaged adipocytes, and it appears large, white, round, surrounded with loose connective tissue. So, I gave you a big hint there. It's adipose connective tissue. Next, in the middle, there's a few words that kind of give it away, but these are the labels for the reticular fibers, so we can go ahead and assume that this is reticular connective tissue. But some of the things that give it away are its very spotty and regular appearance. So that spotty appearance is found in a lot of organs. So we see it in the spleen, the lymph nodes, and bone marrow. Next on the right, it kind of looks like a lot of stuff is going on there, kind of like cellular confetti. But it serves as a universal packing material between other tissues. So it's got a lot of elastic fibers in there, provides cushioning, and it plays a role in inflammation and tissue repair. So hopefully that gave you some hints. It's areolar connective tissue. Okay, I hope you're taking notes because there might be some test questions with this tissue and images. So on the left here, I'm just going to tell you what these ones are because most people get these right. And that is simple squamous on the left, so that it's harder to tell the difference between adipose tissue and simple squamous. But I'll tell you right now, there are layers here. You see there's not just one cell. So I'll go back so you can, well, you can go back. So you can see there's a bunch of cells lining this one pocket here. This is simple squamous tissue, and it lines blood vessels and the alveoli of the lungs. It's really good at efficiently diffusing stuff like gases along the tissues there. Next, you see keratinized stratified squamous, and then on the right is non-keratinized stratified squamous. The way you can differentiate these is this very tightly packed dark staining area has a very dark layer on the very top, and that's the keratinized stratified squamous. So stratified means there's many layers, squamous means irregular shapes, right? And so we have our keratinized here, which is the skin. You just like look at your hand, you're looking at keratinized stratified squamous. And then there's non-keratinized stratified squamous, which is more similar to the inside of your mouth. All right, so now let's take a look at this image on the left. You see these regularly shaped, regularly spaced, cube-shaped cells surrounding these lumens. Those are really found in a lot of glands and ducts, and they're really good at secreting stuff and absorbing stuff. So these are called simple cuboidal epithelium. Simple meaning there is only one layer. Cuboidal, well, they're cuboidal shaped, and epithelium, they're along the lining of these lumens here. Next, here on the right, you see more column-shaped cells. There's also only one layer there, and they're also accompanied by goblet cells. These are found in the digestive tract because they're very good at secreting and absorbing stuff. So you digest stuff with digestive enzymes, and you need those goblet cells giving mucus to help lubricate that area. So this is simple columnar epithelium. So you see there's only one layer, they are column-shaped, and they are along a lining. The next image here, you see kind of column-shaped. Yeah, I would say that's column-shaped, but they're kind of weaving in between themselves, and they have these hairs there. And those hairs actually move to help bring mucus out of your respiratory tract. So this is lining a lot of the bronchioles in your respiratory tract. And let's see if you know what it's called. It's pseudostratified ciliated columnar epithelium. Now that's a mouthful. Pseudostratified means it's kind of faking being stratified, and that's because they're kind of obliquely intruding on themselves. And then you have ciliated, meaning it has cilia, like these hairs right here, and then they're columnar shaped, and they are along the lining of a lumen, so they're epithelium. All right, there's only one more image after these two, I promise. So this is transitional epithelium, and it's specialized to allow the urinary organs to expand and stretch without tearing. It helps with the adapting fluctuations in the volume of the fluid in those organs, and it's really important for that. Last but not least, we have, anyone know? This is bone. Bone's also a connective tissue. It's very important for structural integrity of our body, for movement. There's also calcium deposits here, so we can store calcium, which is really important for our nervous system and our chemical gradients within our body. Using a microscope correctly is crucial for examining tissues and other small structures. Here are some key points. Carrying the microscope. Always carry the microscope with two hands, one on the arm and one under the base. Setting up. Place your slide on the stage and secure it with the stage clip. Focusing. Start with the lowest magnification, 4x in our case, and use the coarse adjustment knob to focus. Once focused, switch to the higher magnification and use the fine adjustment knob for a clear detail. Cleaning. Cleaning the lens with lens paper and not your shirt or tissue helps to avoid scratches. Observation. Spending time observing each specimen at different magnifications to understand the details of each tissue type. You're going to be making detailed drawings, so it is crucial for you to be able to change between the objectives so you can see all the structures necessary for your drawings. And so, understanding proper etiquette will take you a long way. Dissection is a critical part of learning anatomy. It provides a hands-on understanding of the spatial relationship between structures, and there's a lot of things you need to know to do them appropriately. First of all, treat all specimens with respect. Treat yourselves with respect by using proper PPE. Treat your lab mates with respect by handling the equipment properly. So we use scalpels, we use scissors, these are all sharp, and we don't want to have any cuts. Remember to keep your area clean, both before and after doing the dissection, and make sure you dispose of the waste appropriately. Your TA will walk you through how to do all this. Use precision cuts and follow instructions closely to avoid damaging the specimens. Also, make sure you're not cutting towards your hands. Some people cut a little too aggressively and end up cutting their own hand. Take your time, work methodically. It's really easy to get carried away, but you should also enjoy your time dissecting. All right, before you leave, here are some reminders. Look through the syllabus for required materials and make sure to order those. Look at the due dates for assignments and plan ahead for each lab. If you have any questions, just email your instructor. Their email will be on the syllabus, and you will be getting an email from them before your first class. Enjoy the semester, and see you in class.